Hi, I'm Taj Williamson. This is my presentation on Newt and Garter snake coevolution. Evolution is a process of change in all forms of life over generations, and evolutionary biology is a study of how evolution occurs. Biological populations evolve through genetic changes that correspond to changes in the organism's observable traits. A key component of evolution is population change. In biology, coevolution occurs when two or more species reciprocally affect each other's evolution. Charles Darwin mentioned evolutionary interactions between flowering plants and insects in his book On the Origin of Species. Population change, defined generally, is the difference in the size of a population between the end and the beginning of a given time period, usually 1 year. Specifically, it is the difference in population size on January 1st of 2 consecutive years. Mutation can be defined as a changing of the structure of a gene resulting in a variant form that may be transmitted to subsequent generations, caused by the alteration of a single base unit in DNA or the deletion, insertion, or rearrangement of larger sections of genes or chromosomes. Two species that have been proven to show coevolution are snakes and salamanders, particularly the newt and the garter snake. This newt is a small semi-aquatic amphibian that has developed high contents of dangerous toxin in its body, specifically tetrodotoxin, or TTX for short. The Pterica granulosa is the scientific name of this particular newt that is generally found in the forests of North America. Thamnophis is the scientific name of the garter snake, and this type of a garter snake is a small to medium-sized striped snake that inhabits North America. One a predator, the other a prey, however both adamant to survive. As a result, both the newt and the garter snake have co-evolved in response to the other's evolution, attracting scientists and the world to this phenomenon. This coevolution was discovered by one then undergraduate student, Edmund Butch Brodie Jr. In the 1960s. Brodie and his son, Edmund Brodie III, through their studies, began measuring the rate of evolution in the rough-skinned newt and the garter snake. Butch had an original quest to figure out what had killed some hunters in their campsite, and this started him on a serious journey into evolutionary biology. After discovering that the newts were indeed toxic, he began investigations into why they had evolved such extreme toxicity, and in the process, uncovered a raging battle, an evolutionary arms race. The unfortunate hunters were simply caught in the crossfire. The Brodies had predicted that if snakes and newts are co-evolving, their traits should match up. That is, because TTX production is costly for newts and TTX resistance is costly for snakes, newts should be just toxic enough to avoid predation, and snakes should be just resistant enough to eat a newt. Co-evolving organisms often match traits this way. When the Brodies and student Ben Ridenhour compared snake resistance and newt toxicity in different populations of snakes and newts, they found just what they predicted, a close match. Where low toxicity newts occur, the snakes in the area have low resistance. Newts of intermediate toxicity occur with snakes of intermediate resistance, and in some populations, the Brodies discovered extremely toxic newts that were being eaten by snakes with extreme resistance. He found that newts that produced higher levels of TTX had less energy to produce future offspring, while for the garter snakes, those that were more TTX resistant slithered at a slower rate than those snakes that were non-resistant to TTX. Tetrodotoxin kills by quirking molecular pores on the surface of nerve and muscle cells, which act as channels for sodium ions. If these ions can't traverse the channels, muscles can't contract and nerves can't fire. As a result, the induced creature becomes paralyzed and ceases breathing, thus leading to death. The garter snakes are able to prevent this because they have developed a mutation in which they change the shape of their sodium channels, so the toxin is unable to plug them. In addition to this finding, researchers from the University of California, Berkeley, have found that in the areas of North America where there are newts with lower toxin levels and thus higher reproductive capabilities, there are garter snakes present with lower resistance to the toxins and therefore faster moving or movement. This graph illustrates the effect TTX has on snakes that consume rough-skinned newts as it relates to the speed of their movement after consumption. It shows that the snakes that reside in areas with newts without the toxin tend to consume less TTX and the speed of their movement remains more or less intact, as opposed to those that consume highly toxic newts. Their speed is slightly reduced. The coevolution shared between the newts and the snakes led to Dr. Brody and other scientists to generate the arms race analogy. This basically means that as one creature develops methods that enhances its ability to survive or combat the other, in response to this, the other creature mutates and adds to its firepower. Simply, as a newt becomes more toxic, so does a snake become more resistant, ending in an arms race. It is believed by many scientists that the snakes actually have the advantageous hand in the race, as most snakes were able to still consume toxic newts, though they are not as resistant as other snakes. Charles Hannifin posited and proved this through research samples where he found that in a third of the locations sampled down to the least resistant snakes to TTX were capable of consuming the newts with high levels of toxin with little to no problems, as the consumption of the toxic newts hardly affected their movement. The arms race, however, is not completely present in all cases. In Brody's research, he found that not all garter snakes were resistant to the toxicity of the newts, leaving question marks of the true advantages of snakes in the arms race. Here's a quote from Brody, 1968. Brody reports a Tarica granulosa that was eaten by a bull. Brody reports a Tarica granulosa that was eaten by a bullfrog later emerging unscathed from the dead frog's mouth, and we have removed live newts from non-resistant garter snakes that died up to 85 minutes after